The nervous system controls and integrates the activities of the different parts of the body. It is divided into two main parts, the central nervous system, which consists of the brain and spinal cord, and the peripheral nervous system, which consists of a paired series of cranial and spinal nerves and their associated ganglia. Functionally, the nervous system can be further divided into the somatic nervous system and the autonomic nervous system. The main difference of the somatic nervous system and the autonomic nervous system is that the somatic nervous system acts upon the body's external environment primarily through the actions of the skeletal muscle and uses mostly voluntary responses to consciously perceive sensory signals from the body wall and limbs. The autonomic nervous system acts upon the body's internal environment and is concerned with motor control of smooth muscle, cardiac muscle, and glands, and uses mostly involuntary responses to sensory signals that are not consciously perceived. The hypothalamus of the brain controls the autonomic nervous system and integrates the activities of the autonomic and neuroendocrine system. Therefore, the autonomic nervous system makes a significant contribution to homeostasis. Some of its actions include regulation of blood pressure, gastrointestinal responses to food, contraction of the urinary bladder, focusing of the eyes, and thermoregulation. The autonomic nervous system is divided into two parts, the sympathetic division and the parasympathetic division. The sympathetic and parasympathetic nervous system have different effects on the body. The sympathetic nervous system, or SNS, controls functions like increased heart rate and blood pressure, as well as slowing digestion, all of which maximizes blood flow to the muscles and brain, which can help you either flee from a threat or to fight, otherwise known as the fight or flight response. In comparison to the parasympathetic nervous system, the sympathetic preganglionic fibers are often closer to the spinal cord, while the parasympathetic preganglionic fibers tend to be longer and synapse with the postganglionic fibers, which are situated closer to the target organs. Preganglionic fibers T1 to T4 at the thoracic region enter the corresponding ganglia of the sympathetic chain, and synapse at three higher level ganglia, the superior, middle, and inferior cervical ganglia. At the superior cervical ganglion, postganglionic fibers exit to innervate the lacrimal glands, the radial muscles of the irises, and the salivary glands. In the eyes, it stimulates pupil dilation, causing far vision, increased salivation by salivary glands, lacrimation of the lacrimal glands, in addition copious sweating by the sweat glands and contraction of the piloerector muscles when stimulated. The inferior cervical ganglion usually fused with the first thoracic ganglion to form the stellate ganglion. Together, the middle cervical ganglion innervate the heart, lungs, and bronchi, which results in increased heart rate through increased contraction of the cardiac muscle. In the esophagus, motility is decreased, and at the bronchi is bronchial dilation. Preganglionic fibers T5 to T9 form the greater splanchnic nerve and synapse with the prevertebral ganglion known as the celiac ganglion. It exits as a postganglionic fiber to innervate the stomach, liver, gallbladder, pancreas, and small intestine. In the stomach, it decreases absorption and inhibits peristalsis and digestion. Glucose release at the liver, while stimulation causes gallbladder and the bile ducts to relax. At the pancreas is decreased production of insulin. Greater splanchnic nerve also synapses with the superior mesenteric ganglion and innervates the intestine, decreasing peristalsis, absorption, and secretions. Furthermore, at the adrenal medulla, postganglionic fibers synapse with the chromaffin cells, releasing norepinephrine and epinephrine in response to stress. T10 and T11 form the lesser splanchnic nerve, and together with the T12 synapse with the aorticorenal ganglion and innervate small intestine and kidney. Stimulation of kidney results in decreased urine output and increased renin secretion as well as epinephrine. It is further emphasized that the kidney is stimulated only by the sympathetic division. L1 and L3 synapse the inferior mesenteric ganglion, supplying different parts of the colon and upper rectum, all of which result in decreased peristalsis. Furthermore, it passes through a hypogastric plexus and constricts the urinary sphincter. In addition, the gonads is stimulated, promoting emission prior to ejaculation for males, while in females stimulates contraction of smooth muscle. The parasympathetic nervous system generally acts as an antagonist to the sympathetic nervous system. In other words, it controls bodily functions at a state of rest and conserves energy. The parasympathetic or craniosacral division has preganglionic fibers that pass uninterrupted all the way to the organ, and the postganglionic fibers are located within its walls. While both sympathetic and parasympathetic preganglionic neurons are cholinergic, almost all of the postganglionic neurons in the parasympathetic are also cholinergic. Thus, terminal endings typically secrete acetylcholine. The parasympathetic division covers the cranial region and intermediolateral nuclei of the sacral spinal cord. Originating from the central nervous system are Edinger's westphal nucleus, superior salvatory nucleus, inferior salvatory nucleus, nucleus ambiguus, and dorsal motor nucleus of the vagus. The cell bodies in the Edinger westphal nucleus of the ocular motor nerve, otherwise known as the CN3, project to the ciliary ganglia to innervate the ciliary muscle, which causes contraction for near vision, and pupillary sphincter of the iris that contributes to the constriction of the pupil. The superior salivatory nucleus is in the rostral medulla and contains the parasympathetic neurons that project through the branch of the facial nerve, CN7, to the pterygopalatine ganglion. The postganglionic fibers supply the lacrimal, nasal, and palatine glands, in which increases the rate of secretions. Another branch of the facial nerve carries preganglionic fibers to the submandibular ganglion, in which supplies the postganglionic fibers to the two salivary glands, the submandibular, also known as submaxillary, and the sublingual glands. The cell bodies of the inferior salivatory nucleus of the glossopharyngeal nerve, or CN9, project to the otic ganglion to innervate and increase the rate of secretion of the third salivary gland, known as the parotid gland. About 75% of all parasympathetic nerve fibers are in the vagus nerve, or CN10. Cell bodies of the vagal preganglionic parasympathetic neurons are found in the medulla within the nucleus ambiguus and the dorsal motor nucleus of the vagus. Neurons in the nucleus ambiguus innervate the sinoatrial and atrioventricular nodes in the heart, which will decrease heart rate and blood pressure and stimulates the contraction of striated muscles in the pharynx, larynx, and upper esophagus due to the activation of somatic motor neurons. Stimulation of the dorsal motor nucleus of the vagus induces many effects in the viscera, including the initiation of secretion of gastric acid, insulin, and glucagon. Sacral parasympathetic fibers travel through the pelvic splanchnic nerve to their terminal ganglia and are distributed to the descending colon, rectum, bladder, and the reproductive organs of both males and females. In the colon, specifically the sigmoid, descending, and distal half of the transverse colon, motility is increased through peristalsis. At the rectum, the internal anal sphincter relaxes, which allows defecation. In addition, the detrusor muscle is stimulated, resulting in bladder contractions, as well as relaxation of the internal urethral sphincter. While in the gonads, nitric oxide is released, which dilates blood vessels to increase blood flow, causing an erection in males, and in females it causes a clitoris engorgement. The enteric nervous system is a collection of nerve plexuses that surround the gastrointestinal tract, including the pancreas and biliary system. The number of neurons in the enteric system is greater than 100 million, more than the number in the entire spinal cord. So this highly developed enteric nervous system is especially important in controlling the gastrointestinal movements and secretion. The enteric nervous system is composed mainly of two plexuses, an outer plexus lying between the longitudinal and circular muscular layers called the myenteric plexus or Auerbach's plexus, and an inner plexus called the submucosal plexus or Meissner's plexus, which lies in the submucosa. In the intestinal wall, the myenteric plexus is involved primarily in the control of motility, whereas the submucosal plexus is involved in the control of ion and fluid transport. Both the myenteric and submucosal plexuses receive preganglionic parasympathetic innervation from the vagus nerve. And that sums up our autonomic nervous system. And on behalf of the SGD4, thank you for listening.